Alright, AMP1 students, in this video we're going to continue with Chapter 3 and we're going to talk specifically about enzymes. And we're going to take a quick look at our objectives so you know where we're at. So in the last video we talked about what an enzyme is and we talked about activation energy. So we're going to talk a little bit more about the enzymes and what their structure is and what their functions are and where you can find them. So that's in these learning objectives right here in the center. So, alright, so we're going to go to our textbook and we're going to look at the definition again of enzymes, just to remind everyone that an enzyme is the molecule that's going to catalyze chemical reactions. So that just means that they're going to make chemical reactions happen faster. So that's what the enzymes are. That's their purpose in our bodies, is to make chemical reactions happen faster. So they use that term catalyze to describe something that accelerates a chemical reaction. All right, so we're going to, that's their function. I'm going to slide down here and we're going to look at their structure. So when it comes to an enzyme, its chemical structure is, it's a macromolecule, organic one, and it's a protein. So it has all those amino acids as the building blocks, and they all fold into very complex structures until we get this large, complex, specifically shaped molecule. So this little blue thing in this picture is showing one possible enzyme, some generic enzyme in our body. So that's the structure of an enzyme. If you remember when we talked about proteins in Chapter 2, we mentioned that one of the functions of proteins is they serve as enzymes. Some other functions where they could serve as a hormone, or they could be like the muscle proteins, actin and myosin. They could be hemoglobin. So they got a lot of purposes in the human body. So one of the purposes of protein is they serve as enzymes. So all the enzymes in the body are chemically proteins. So they have that carbon, hydrogen, oxygen, nitrogen. Those are their 4 atoms. And they've got the building blocks, which are amino acids, but then they fold, and hydrogen bonds kind of help hold them into a specific shape. Other bonds do too, but we talk mainly about hydrogen bonds. So that is the structure of an enzyme. It is a protein, and it's going to have a specific shape, as you can see this one does here. Okay, so since we're looking at this picture, we'll talk about the specific components of a enzyme. So you'll notice with this enzyme, there's this one area, they call it the active site. So the active site is, and it's going to be like a specific shape. And so this enzyme has this specific shape of its active site. But then if we have another enzyme, it'll have a different shape of its active site. And so the active site is the site on the enzyme where we call it a substrate. So where the substrate binds. And the substrate is just whatever reactants or products that this, actually it's reactants, whatever reactants that are the proper shape to fit into the active site of the enzyme. So it's these enzymes, so structurally they're all proteins, and then another characteristic of them is they're very specific. So they have this specific shape, so the only chemical reaction that they can catalyze or speed up is the one where the substrate or reactants of the chemical reaction match the shape of the active site and can fit in there nicely. So in this case, this little substrate, you can see the way it's shaped, it's going to fit perfectly in there. And then we'll get this little enzyme substrate complex, just means that the enzyme and substrate bound together. So those are the structural components of an enzyme. So it has an active site, and then the substrate is whatever particular reactants for that chemical reaction that this enzyme is specific for. So that's why we have thousands of enzymes, because we have like thousands of chemical reactions in the body, and you can't use each enzyme will only catalyze one, in general, one particular chemical reaction. So we have to have, thousands of differently shaped enzymes in order to catalyze all these chemical reactions going on in the body. So this is a good image to show the structure. But the book also uses this image as well. So it shows this active site, kind of looks like two little scoops in it. But again, it's like the right shape for the substrate. So, and then this one looks a little bit different. So it's got an active site here. And so the little substrates can fit in there perfectly. So, all right. So that is our anatomy or structure of our enzymes that we need to be familiar with. All right, so let me go to our objectives. Okay, so we talked about the structure of an enzyme and the general functions of enzymes and the key structural components. Before we get to the steps that happen when an enzyme catalyzes a chemical reaction, we'll talk about where you can find enzymes in the body. So enzymes can be found inside cells. So sometimes the enzymes, it'll be made by the cell, and they will stay inside the cell. And then there's certain chemical reactions inside the cell that they will need to catalyze. Now some enzymes will actually be placed or embedded in the plasma or cell membrane of a cell. So then it will not be outside the cell, but not really be inside the cell. It'll be in the plasma or cell membrane. And then because of its location, things can come and bind to it, and then its specific substrate can come and bind to it, and then it can catalyze chemical reactions that way. And then the third place you can find enzymes is outside the body. So some cells will make enzymes, and then they will, through exocytosis, they'll release them. And so that's pretty common in the digestive system. We have our cells, epithelial cells that line our small intestines, and so they may make the enzyme lactase, and whenever they make that enzyme, then they'll release it out into the lumen, which is the area where the milk or any sort of food might be. I say milk because lactase is going to catalyze the lactose, the chemical reaction, the digestion of lactose, which is the sugar that's in milk. And so you can have enzymes that are made in the cell, but then they are released from the cell, and then they do their catalyzing chemical reactions outside of the cell. So, all right. Okay, now we're going to go to our picture showing the steps in when an enzyme catalyzes a chemical reaction. And I believe, so we have a picture here, and then, which I'll probably use, but then below we have the listed steps that occur when an enzyme catalyzes a chemical reaction. So let me increase this so we can look at it a little closer. Okay, so the first step that will happen when an enzyme catalyzes a chemical reaction, you have to have the binding of that substrate to the enzyme. And when that happens, then what is formed is called an enzyme substrate complex. So here's our enzyme, here's our substrate. The enzyme will bind, I guess the substrate will bind here to the active site on this enzyme, and we get this enzyme substrate complex. So that's the first step. And then the second step is the enzyme is going to kind of hug the substrate a little bit, and it'll kind of change shape. And so that just allows for a nice tight fit between this enzyme and substrate. They call it an induced fit between the enzyme and substrate. And that's important because what that does is when it kind of hugs it, it stresses that bond, and we want to break that bond. So this is, stressing that bond there, and then it will break. So that's kind of showing the breakage of that bond between those two sugar molecules. And then after that happens, then the bond is broken. The enzyme will release the substrate, and so these are our two little sugar molecules that are products from this decomposition reaction, is what this is showing. And then the enzyme is going to then go away, and it will find another molecule, substrate molecule, lactose in this case, and then it will catalyze that chemical reaction of decomposition reaction of lactose in this case. So it doesn't, once this enzyme, once it catalyzes a chemical reaction, it releases its products, or the substrate, and then it will go and find another molecule that fits, its specific molecule that fits, or substrate. And then so it can continue to catalyze a chemical reaction over and over and over again. Now eventually it's a protein, so eventually it'll wear down and it'll be broken down, and then the cells will have to make new ones. But it doesn't just it doesn't just catalyze one chemical reaction. It can catalyze multiple chemical reactions in a certain period of time. So, all right, so that's another characteristic of enzymes, is they're not destroyed after they catalyze a chemical reaction. They can catalyze a chemical reaction over and over again. All right, so let me go to our objectives. So we talked about the structure, the function, the components, where you can find them in the body, and then the steps, and then the naming of enzymes. Now this is, find the key here. Well, actually this one kind of shows it. They usually, not always though, but usually, because the enzymes are specific, they have a certain shape, and their active site has a certain shape, they will only catalyze or bind to a specific substrate. So it's really easy. If we know this enzyme will bind only to this substrate, then what they do is, we're familiar that this is the substrate, lactose. So then they just name the enzyme that only binds lactose, lactase. So usually it's the ASE at the end that indicates that it is an enzyme. So not all enzymes end in ASE. We'll see some of those that don't, but a lot of them do. So if you ever see something that has ASE at the end, it's likely a enzyme, and then you can usually figure out from the first part before the ASE which substrate it actually is going to bind to and catalyze a chemical reaction. So, all right. So I'm going to, because the next objective, we are going to talk about enzymes and how temperature and pH, and then we'll also talk about inhibitors of enzymes. So we'll do that in the next video because we won't have time in this one to finish that. So, all right, I will see you all in the next video.